Thank you. Thank you for the introduction, and also thank you to Bruce Fischel for inviting me to give a talk here. So it's really nice to be back in Boston, and so today I'm going to describe some of the work that's happening in my lab for sort of tackling different clinical and neuroscience problems, and also kind of describe the different modeling frameworks and strategies that we're using to tackle the challenges of the data sets that we have access to in this space. And so the main theme from a modeling standpoint is the idea of bridging kind of old school machine learning techniques with kind of new current trends in machine learning or AI, which involve deep learning. Right? So just to set the stage a little bit, I wanted to give just a little bit of information about my motivation for tackling these problems. And it's just a slide of statistics. So by the end of the year, there'll be about 1.25 million children in the United States living with autism. About 49,000 young adults would have had their first schizophrenic break. There'll be almost 800,000 incidents of stroke. 16.2 million people will go through a major depressive episode, and 5.5 million elderly adults will start to manifest the early signs of Alzheimer's. And so the purpose of this slide is just to illustrate that disorders of the brain are pervasive. So they're very common in society, and they represent collectively a massive sort of public health concern and initiative. And it's also to highlight that these disorders, they really occur across the lifespan. So from early childhood to late adulthood, there are issues and people who are suffering and kind of looking to us to come up with new solutions. And so where do I fit into this scheme? So I'm not a neuroscientist or a biologist by training. So my training is more in signal processing and data analytics. And so at least my contribution I see is to look at this from a data mining or a data science point of view. And so a lot of the work in my lab is bringing together these tools from artificial intelligence and using them to integrate multiple sources of information and also strategically blend sort of a priori clinical insights or structured assumptions about the data themselves to do better biomarker discovery or therapeutic planning and outcome assessment. So these are just 3 stages in the clinical care pipeline, and we really want to address all of them depending on the problem domain and what makes sense. Now, unlike many applications that are using AI techniques, so vision, computer vision is one of them, and speech is another, we have a lot of unique challenges in this space of sort of neuroimaging data, functional neuroimaging data for clinical neuroscience applications. So the challenges can be divided into small data sets and complex phenomena. So on the data size, so medical imaging in general, we tend to have smaller data sets, and it's even worse with functional imaging modalities and as you move to clinical populations. So where a computer vision application might have 100,000 or million images to learn from, many of the data sets that I'll be talking about today, we have on the order of 10 to 50 patients. So that's a very reduced sample size, and it's really not amenable to out-of-the-box techniques that have been developed. Another factor that causes these small data sets is that, especially with the functional modalities, there's a great deal of confounds in the data. And those confounds are actually aggregate across, they're very site specific, essentially. So if you try to aggregate data across sites to get larger databases, what you end up with is just very noisy site variability which tends to dominate your analysis. And so in some sense, it constrains your problem in many cases to be small. So on the flip side, we do have a lot of complexity. So if we're looking at disorders of the brain, these disorders tend to be very subtle, especially in cases of such as autism, schizophrenia. And so we are trying to find these very subtle differences with very small data. There's also patient heterogeneity, which many models actually ignore. So this heterogeneity, again, makes it very difficult to fit a comprehensive model. And unlike sort of computer vision or speech, where we have a good idea of how to parse these images, in sort of functional neuroimaging, we don't really have a ground truth. So there's really no accepted way of assessing, okay, we found a phenomenon or we found a biomarker, how do we evaluate if it's correct or not? And so I guess sort of most of my research is kind of trying to tackle different elements of these 2 challenges and using different modeling and analysis techniques to do so. So this is the outline for the remainder of the talk. And so I'll be talking about 3 different projects going on in the lab. And I picked these projects because they represent a range of modeling techniques, as you'll see, a range of clinical disorders, and also kind of a range of what is the point at which we are trying to tackle the problem. So from basic research to therapeutic planning to a very far out there project. So I'm going to start with the project that is closest to my bread and butter, and it looks at functional connectomics. And so our goal in this project is to predict clinical severity. So for those of you who might not be as familiar, we're working with resting state fMRI. So fMRI in general measures the brain dynamics. You can think of it as a 4-dimensional acquisition. So there could be a collection of volumes over time, or an alternative perspective is each spatial location in the brain has an associated time series. Now, in terms of resolution, spatial resolution is considered to be quite good, between sort of on the order of millimeters, but the temporal resolution is very, very slow, and so it's much slower than neural activity. Now, as I mentioned, we're looking at a resting state acquisition. So unlike sort of a task-based paradigm, so in resting state, the patient is lying very quietly in the scanner, and the measure of interest is not a particular activation, but looking at these pairwise relationships or kind of co-activation patterns across the brain. So our feature that's input to the model are these row ij's, so the correlation between the time series at spatial location I and j, so the purple and the green signals in the slide. Now, the reason that this has come to be a biomarker is because over about a decade or so of research, it's been shown that regions which reliably co-activate are likely to be involved in the same functional processes. And so on the slide, I've just illustrated 3 well-known functional systems, the motor, default, and visual, that were identified using a seed-based analysis, so having high correlation with a particular seed. Now, we can take this seed-based analysis one step further and look at all pairs of correlations between all regions of interest or voxels of interest in the brain. And this leads us to a whole brain analysis. And sort of the input here are these symmetric correlation matrices that capture the pairwise correlation between all points of interest. Now, in terms of prior work, so this correlation matrices have been kind of a starting point to much of the literature, and there's different layers of analysis that have been developed. So in the simplest case, one would run maybe a statistical test to compare features of these correlation matrices between patients and controls. And so here, from a mathematical standpoint, what we're looking at is features in which the distribution of these measures are different in patients and controls. In the simplest case, we might do a univariate test on an individual entry of this matrix, so an individual edge. Alternatively, graph theoretic metrics such as hubness or centrality have become very popular, and this can be another input feature. Just a plug for some of my prior work, we had developed a sort of a Bayesian network modeling that tried to do this in a holistic fashion to identify targeted hubs of different neurological disorders. But I think the unifying theme of this first half of the slide is that these methods are all designed for a group-based analysis, kind of trying to explain what is fundamentally different between a patient cohort and a control cohort. If we think about translating this to make patient-specific predictions, here machine learning has really been king. And so at a very high level, you can think of most machine learning methods as these 2-stage pipelines. So you have resting state fMRI data coming in, typically a correlation matrix. You do some amount of feature extraction. So this could be as simple as the topological measures. This could be some sort of projection onto a manifold. And then from those features, you build a predictive model, typically a classification model, and you receive an output for a patient. Now, this pipeline has been very common and very successful in the context of binary classification. So taking resting state fMRI and predicting, is this a patient or is this a control? But it has had less success if we want to sort of characterize any finer grained assessment of the patient. So potentially clinical severity or behavioral deficit, et cetera. And so the way we're approaching this problem of the continuous characterization is by coupling this feature selection and predictive modeling block into kind of an aggregate framework that is both interpretable and gives you some predictive value, and it can learn from small data sets. So here is kind of the model overview for this particular project. So this is work done by my graduate student Shimona at Johns Hopkins and our collaborator Stuart Mostovsky at the Kennedy Krieger Institute in Baltimore. So as you can see, our model takes on two different terms. So there's a data representation component, which is this blue L-shaped box. And so that data representation is what gives us interpretability. And so from these collection of similarity matrices across the patient cohort, we're going to extract some group level information that is interpretable, that can be projected back onto the brain, and some patient specific information that will then feed into our regression model, which is the green box shown. And so I'm going to build this up in a little bit more detail for you subsequently. So just starting with this L-shaped data representation term, so we have patient correlation matrices, and we're going to assume that we can decompose these correlation matrices into a sparse subset of elementary basis or basis subnetworks. So a basis is kind of represented by a single column of this matrix B, and it's essentially each column is a heat map that you can project onto the brain, and it tells you template or exemplar patterns of coactivation that is common or that is very explanatory or has high explanatory value throughout your data set, right? And so to match dimensions, remember we have a correlation matrix. We're going to use an outer product representation. So there's a B and a B transpose to get essentially a matrix representation. And then there's this diagonal weighting matrix C. And so the elements on the diagonal are really the contribution for that particular subnetwork to that patient, right? And that's what distinguishes kind of across the patient cohort, right? And so subbing this back into our overall framework, these basis matrix, the basis matrix B, each column will give us the representative subnetworks, and these patient coefficients are what we are going to feed into the regression model. And so we can think of, now we have the interpretability from the data representation box, so we can think of doing this regression model as kind of increasing level of complexity as we need to. So our first iteration was just a very simple multivariate linear regression. And so this is work that appeared at McKay in 2018. And so this joint optimization actually gave us the only thing that we could develop, well, the first thing that we could find that would fit, that would generalize in terms of being able to predict clinical severity on new patients. Now, from here, we can, again, think of building layers of complexity, and the next one is doing patient embedding. So this is very similar to kernel ridge regression. It was work that appeared at IPME earlier this year. And this, it was a very interesting mathematical framework, but it also gave us robustness to parameterization, or the hyperparameters of the model. And then we can think about even extending this further to do more complex tasks using neural networks, right? And so we can consider the regression model itself as just being a neural network where the input is that the coefficients on the diagonal and the output is what we care about clinically. And this appeared at McKay just recently, a few months ago. And what we were able to do here is multi-score prediction, so characterizing multiple dimensions of a particular disorder at once. Now, just to dive beneath the hood a little bit, in terms of the mathematics, so we have this joint objective function, which has unknown quantities of the bases, the patient coefficients, and then whatever the parameters of the regression model are. So that these are two terms. So there's the matrix factorization on the left, which has, you can see that kind of outer product formulation. And then there's we're using an artificial neural network, but it can be an arbitrary neural network as to your specifications, but it has a loss, right? So this is the prediction term. Now, so right now, and for this particular work, for the multi-score, we're just using a very simple mean squared error loss function across all three clinical scores. We have tried a little bit more complex functions, so doing bimodal distributions. You can think about even using the same formulation to do classification by specifying like a cross-entropy loss there. Now, in terms of the overall objective, it gives us the two components, but it is ill-posed, and so we have some regularizers. So the first is we impose sparsity on the bases, and so essentially each basis we want to say captures sort of interactions between a sparse set of regions, so it isn't kind of the entire brain, and it's a little bit easier to tease apart interactions. And then we have a ridge penalty on the coefficients, and this is primarily for convergence of the optimization. So this, if we look at the formulation, all we actually have as input during training are the matrices gamma, which are the correlation matrices, and the scores y, which are whatever the behavioral paradigm or clinical paradigm that you're administering. So the B, the C, and the W, or the parameters of your ANN, are actually unknown. And so in order to do this, to estimate them, we're going to use an alternating minimization framework, where essentially we're going to fix two of those components and update the third one. So the update for the matrix B, so the bases, is done by proximal gradient descent because of the L1 penalty. There's an LBFGS update for the coefficient C. So this is an optimization routine that was developed previously. And what's really interesting is that routine actually is what couples the two terms. So you have essentially some derivative from some gradient from the matrix factorization, as well as a backpropagated gradient from your neural network, and that sort of couples the two representations. And then we update the neural network using whatever your favorite optimizer is. So we use Atom in this case. That was a typo. Yes, they should be lambda 1 and lambda 2. So if we look at training and testing, so during training, we have a bunch of correlation matrices and a bunch of clinical scores that correspond to a single patient, to those patients. We're going to use that to estimate and fix the bases and the neural network. And now once we do this for a new patient, we have to figure out what their coefficient, their C, is. But since we don't know what their clinical score is, we'll just use those three terms to estimate the C's, and then feed that through the neural network to obtain an output prediction. The neural network we use in this case is a very simple two-layer ANN, two hidden layer ANN, with 8. So the input is the dimensionality of the bases, which is 8 in our case, and then the output is we have 3 scores for autism. So the autism data set we're looking at, and that's the output of the network. So we did a preliminary validation of this framework on a data set of 52 autistic patients recruited at through Kennedy Krieger Institute in Baltimore. So it's a region-based analysis. So we use the AAL atlas to subdivide the brain into 116 cerebral, cerebellar, and subcortical, and cerebellar regions. And then our features are the correlation matrices that were constructed between the mean time series of the two regions. It's a very standard pipeline. We have looked at other parcellations as well, and this formulation is agnostic to it. In terms of the clinical scores, we have 3 scores of interest. The first is ADOS, total ROS score. So this is the Autism Diagnostic Observation Schedule. It is actually a kind of a behavioral exam that a trained psychologist will administer to a child. And the dynamic range of this score is between 0 and 30. Then we have the SRS, total raw score, so social responsiveness scale. This is actually a parent report. So it's a written questionnaire that you would give a parent or a teacher or a caregiver, and based on their responses, you compute some sort of severity of social deficits and social functioning. And then that has dynamic range for the autism patients between 70 and 195. And then the praxis is the third measure of interest. And this is a paradigm that's used by our collaborators at KKI. And what it measures is essentially deficits in motor functioning. So in being able to gesture and imitate gestures. And our collaborators are very interested in motor functioning because they've shown, and it has been shown, that children with autism often exhibit motor deficits that parallel their social deficits. So they have problems with hand-eye coordination and imitation, etc. And for validation, we use tenfold cross-validation. So here we'll use 90% of the data for training and then evaluate on the remaining. For comparison, we have our previous model, which is the linear that we had developed 2 iterations past. This was with the linear predictive model. And then we're using BrainNet CNN, which is work that came out last year or a couple of years ago, which is a convolutional end-to-end deep learning method specifically designed for correlation matrices. So here's actually a sort of a grid plot of the results. And so just to parse them, we have each method going down a column. So ours is on the left. Then we have the previous iteration with the linear model, and then the BrainNet CNN on the far right. There's training and testing, and then the metrics are kind of embedded in the plot to read. So first, what we notice is that overall performance isn't perfect for anything, which is actually just a testament to how hard the problem is. But the main thing to note is that all three methods do fairly well and very consistently for the ADAS prediction. So they predict one score very well. But the other two methods, when they try to simultaneously predict other manifestations, it completely fails. Essentially, somehow they don't have the richness or they don't even have the regularization to be able to do that. Whereas our method at least gives very similar predictive performance, predictive trends for each of the three methods. And this is for clinical scores with vastly different dynamic ranges. In terms of the interpretability, so if we project these 8 subnetworks that we estimate in B onto the brain, this is what we get. And these are heat maps. So there's hot colors and cold colors. So the way to interpret it is that the hot colors in a particular plot are all positively correlated with each other. And then regions with colder colors are all positively correlated with each other. And then the hot regions and the cold regions are anti-correlated. And so from this plot, we have a few regions that illustrate kind of default mode and motor cortex anti-correlation. This is very commonly reported trend in autism, as well as visual processing and central executive control, sort of anti-competing contributions, which again, it's a measure that our collaborators are interested to explore further because the visual processing and the motor integration is something that they are actually focusing their research on. So going back to the outline, yes?
Sorry, what was the age range of the subjects in the study?
8 to 12. 8 to 12, yes, so pre-adolescent. So going back to the outline, the first project was much more along the lines of basic computational neuroscience research. The focus was biomarker discovery and maybe some predictive modeling that can be used to either inform studies or better understand autism. The second project focuses on a different disorder, and from a translational standpoint, it actually is more targeted towards a specific question. So the application here is epilepsy, and specifically refractory epilepsy. So epilepsy is one of the most common neurological disorders worldwide. It affects about 1% of the population. And there's an incredible rise in mortality because of this condition. Now, the reason it's actually a very important clinical problem is that between 20 to 40% of epilepsy patients don't respond to drugs, so anti-epileptic drugs, and they continue to have seizures, which is very disrupting to quality of life. And so if we target that, think about that patient cohort, in order to develop any other type of therapeutic strategy, it's crucial for clinicians to understand where and when the seizures are happening in the brain. So where are they coming from and when are they happening? And so EEG is the most important diagnostic tool for seizure detection and localization, primarily because it's the first modality that someone would acquire in the clinic as these patients are going in to be treated. It's also very cheap and very, very prevalent. And what's very interesting here is that despite actually quite a bit of data that you can collect across patients for EEG, and despite this advanced sort of rise of AI type algorithms, almost all of the detection and localization is done manually by visual inspection. And kind of what I mean by that is essentially a neurologist would stare at EEG recordings like this, and they will carry on for potentially hours at a time, and then they will manually decide where on the recording they think a seizure is starting or coming from. And so the second point of the slide is to highlight actually why this is so challenging for AI algorithms. So if I, as a layperson, were to know that, okay, there's a seizure starting on this slide somewhere, I would naturally focus on kind of the noisy area in the middle, which is high amplitude, or some sort of atypical signature kind of on the right of the slide. And unfortunately, my layman intuition would be wrong. So these are muscle artifacts and eye movement. So these are just artifacts of the data that you would see during baseline. And in fact, the signature that the neurologist will pick out is here. And you can see that just the gross contrast between the two and why just naive machine learning algorithms fail. It's essentially because an AI algorithm is going to pick up on the most salient features of your data first, and the most salient feature of your data is actually wrong. And another challenge is that these seizure signatures are different across different patients. They could even be different across seizures for the same patient. And for across patients, they will occur in different channels. Right? And so this is a very complex and difficult problem. Now, if we are looking at still in the data analytics field, there's been quite a bit of work done for automated seizure detection from these EEG recordings. And similar to the previous project that I described, it follows this very standard pipeline. So there's a raw EEG. These methods will then do some amount of pre-processing. And then there'll be a feature selection, so taking these EEGs and trying to extract informative components from them, and then a classification of whether a particular potentially short time window is a seizure or a baseline. Now, I would say the majority of work over the past 2 decades in seizure detection have focused on the feature selection component. And there's been a slew of features that have been explored. So spectral features, time frequency features using short time Fourier transforms or wavelets to try and capture different powers, or even kind of wavelets to capture spiky signatures in the data. There have been nonlinear features, so entropy measures to say, okay, maybe the signals are becoming more chaotic during the seizure and pick that up. But really, sort of these algorithms have kind of fallen by the wayside, and even commercial detectors, which are highly optimized for the task, haven't replaced manual inspection. And so the reason, as I kind of highlighted on the previous slide, is that these are very noisy, very heterogeneous data sets. And so there's plenty of avenue of exploration here. And our approach to kind of tackling this problem is to move away from this pipeline paradigm and to say, instead of making a frame-wise prediction, so trying to extract a meaningful feature and predict just that frame, let's recognize that the actual propagation of a seizure, so the spreading of a seizure, is informative, right? Because we don't expect sort of channels to flip into seizure mode randomly across the head. We expect kind of a steady propagation as it originates. And let's try to capitalize and model that and use that to try and backtrack the onset. And so the modeling strategy we used in this work is based on Bayesian modeling or probabilistic graphical models. And this was work done by my graduate student, Jeff, at Johns Hopkins, and our collaborator, Emily Johnson, in the Epilepsy Center at Johns Hopkins. And so in Bayesian models, we can think of them as divided into 2 chunks. The first is a latent template. So that captures kind of unobserved or hidden phenomenon that we can model, we can inject prior information into, but we don't have direct access to. And then it's complemented by observed data. So in our particular application, this latent template would be, how does the seizure start and spread? So where and when? And the observed data is what we have, the EEG, or features of the EEG as we choose to extract them. So I'm going to start by discussing that top latent component first, and then move on to the likelihood. So the seizure propagation model starts by essentially formulating your graph. So what I've shown is a head model with the electrode channels are in the 1020 system. So this is the standard placement system that is most commonly used in epilepsy centers. And then we've connected them by blue lines. And so these blue lines are a graph we imposed on this topology that connects neighboring channels and contralateral channels. And this actually captures the most commonly observed clinical spreading patterns that you see in the clinic. So kind of locally as well as just right across the hemispheres. Now for the model, we're going to assume that all channels start in baseline. So it's kind of when the patient is not having a seizure. And then as we march on in time, at some point a channel will turn on. So in this picture it becomes red, and that indicates that there's some sort of seizure signature happening in that channel. And again, as we move on in time, that seizure signature will start to spread, potentially first to neighbors, and then it might spread farther than that and even become generalized, possibly. Now, from a modeling standpoint, this is a very common kind of temporal model, and it really mimics a hidden Markov model. So hidden Markov models have essentially a latent state, so seizure versus not seizure, that changes over time. And at every time point, you have an observation. So the latent state is X, and then the observations are Y. Now, the 2 defining features or aspects of the hidden Markov model are the likelihood. So how is the data generated based on the state? as well as the transition. So how do states evolve over time? And so this is, again, based on these two functions, the chain will march along in time. And then given all of the data, we can use something like a forward-backward algorithm to essentially pass messages back and forth and estimate what is the most likely sequence of states. Now, if we translate this back to our particular application, every channel is like an HMM. So every channel is going to transition between these states and has some EEG associated with that. And in our case, we'll have many channels, so one for each in the particular, in the 1020 system. And beyond that, the blue lines cause some sort of coupling between the channels. So now the propagation itself is not just going to depend on its own previous state, but also the previous state of its neighbors. Now, mathematically, this joint distribution of the hidden and observed variables has kind of the observation model, which I'll get back to later. And then the propagation model. So kind of how do these states vary over time? So starting, again, focusing on the propagation model just for a bit, we assume a three-state Markov chain. So there are three states of pre-seizure baseline, seizure, and post-seizure baseline. And what we are essentially construct with the onset and offset probabilities are that channels are more likely to turn on and off depending on the organization of their neighbors. So if we focus on the little gray T5 channel, right now all of its neighbors are in baseline, so it has a low probability of transitioning. Then if some of its neighbors move into a seizure state, then the next time step it has a slightly higher probability of transitioning. And then as more and more of its neighbors are in the seizure state, it has a higher probability of transitioning. Now, going back to the joint distribution, I'm going to defer talking about the observation model for a little bit, just to talk about inference first. But given some sort of likelihood model, inference is actually very difficult in this framework because of all the coupling. It just increases the dimensionality of your state space, and unlike the forward-backward algorithm, it just grows too fast. And so we're going to approximate this posterior with an easier distribution. So we use a mean field variational approximation, where we assume that the true posterior probability of x given y can be approximated by this factorized posterior, where each of these distributions Q is just an HMM, is just a kind of decouples across channels like an HMM. And so the transition and sort of the singleton and pairwise terms on the far right, they're going to depend on the observed data, so the likelihood model, as well as in the previous time step, kind of the expectation of the neighbors in the seizure state. So we've described the prior structure of how seizures originate and spread. And now, in terms of that, remember there was that likelihood model that I deferred. And we can think of, if we look at interpretability versus generalizability, so that top layer gives us interpretability. So once we estimate these states, we have kind of an onset here and potentially a spreading pattern. So we can look at different ways of constructing this likelihood to mine the most informative patterns from the data. So the first iteration of this model, which appeared at McKay a year ago, was just to use a very parameterized approach of, for every time window, we'd use a short time Fourier transform to compute spectral power in different frequency bands, which are fairly commonly used in EEG analysis. And then it was a parametric likelihood of a Gaussian mixture model for each class. But we can even take this one step further and say, even though we only have a few patients and all the patients are unique, we actually have quite a bit of EEG data, so a couple of hours per patient. So we can use all of that EEG data to actually replace the parametric likelihood with a neural network. So we use a convolutional neural network, and here we're training one network per channel to capture different baseline signatures across the head, right, depending on different muscle configurations. Now, there's one small caveat to using a neural network as a likelihood, and that's because a neural network actually, by construction, does the opposite of what we want in a likelihood. So a neural network, we take some amount of data, which is y, feed it through, and it produces a prediction. So this is how you set up a classification network of, in this case, seizure versus not seizure. But what we really wanted was a likelihood, so the flip of that, the probability of y given x, so our data given our state. And so we're going to use a trick of rescaling the CNN output using Bayes' rule to obtain the likelihood. So here from Bayes' rule, the thing we're after, so the likelihood on the left, is actually kind of the CNN output, so the posterior, times the probability of EEG divided by the seizure prior. Now, in our inference procedure, when we run variational, this PEEG, so the probability of Y term, is actually common to all of the states, and so it will drop out, it just cancels. And so what we really care about is that one ratio. And so the numerator of that ratio we have from the CNN, and the denominator we just approximate with the empirical distribution, so the overall probability of seizure versus baseline. Now we evaluate this model on sort of clinical EEG data, 2 clinical EEG data sets. So the first is a data set drawn from Johns Hopkins Hospital. It consists of 15 patients with focal epilepsy. So this actually mimics the assumptions of our data, since in these patients we believe the seizures are starting in an isolated location and spreading. These patients were monitored in the hospital for a few days, and so we have 90 seizures total. The second is a publicly available data set that was released, collected at Boston Children's Hospital and released by John Gutag's group at MIT. It consists of 24 patients and a slightly higher seizure counts, 185, but these are unspecified seizure types. So some of them could be general and actually not as good in terms of fit of the assumptions of our model. And for comparison, we're going to compare to our previous model, which used just a Gaussian mixture model on the likelihood or Gaussian mixture model likelihood on the spectral power, as well as an end-to-end CNN just trained per channel. So in the following slides, or in a few slides, I'm going to show a plot like this. So this is a result plot. What we have is on the y-axis, I've just stacked all the channels. There's no particular meaning in the ordering in which the channels are stacked, so that they're almost like just labels. But on the x-axis is time. So as we move across time, seizures start and stop. And so the black lines in the figure are rough, very liberal onset and offset annotations by a clinician of when the seizure might start and end. And the color is the kind of prediction of each particular model, so the posterior probability. So quantitatively, here are the results for both data sets. So our model is the top PGM CNN. The middle is the previous iteration with the spectral power as the features of interest, and then the bottom is just the CNN. And so there are a few trends to note. First, our model outperforms the others in nearly all of the metrics of interest. Second is that uniformly, all of the methods perform better on the CHB data set. That is because the JHU data set that we have is actually much noisier, and there's less quality control and scrubbing and curation involved. And so I guess the take home here in terms of performance is that our combination of using this prior structure as well as this very flexible likelihood model, so a combination of generative modeling and deep learning, actually outperforms decoupling them. So keeping just the generative modeling versus keeping just the deep learning. Now, in terms of trends that you tend to see, so this is a plot that I had shown earlier for one particular seizure. So we noticed that the assumptions of our model allow us to have continuity. So there are gaps in the predictions for other methods. Now, the CNN is the only method that does frame-wise prediction without any temporal sort of assumptions. And you see that there are a lot of very spurious detections, so it turns on and off very sporadically. And this is probably because the baseline signatures at those particular times were so high that the model picked them up as seizure. And then finally, one nice thing that we get from our model, because we have this prior structure, we can actually look at what it's telling us about the seizure. And so here are 2 patients where the seizure on the right is starting in the right temporal lobe and the seizure on the left is in the left frontal lobe. And the red, or the heat map, is kind of the evolution of our model prediction over time. And you can see that even though the model had no knowledge of where these seizures are starting, and it was not even trained on this localization information, it can automatically infer these patterns from the data in many cases. Now, moving this model forward, while I don't have time to talk about the details, our goal moving forward is to get much better in terms of localization. So rather than focusing just on detection. And so what we showed at MICI this year was the first method that did automated detection and localization from scalp EEG. And so our strategy was actually to decouple the problems into two different layers of the graphical model. And so we used all of the EEG data across channels to infer this, just sort of this top meta chain, which all it did is detected when a patient started to have a seizure. And then once it started, it initiated the spreading model that I just described. And so the combination of the two allowed us to do this localization across many patients in a reliable fashion. So going back to the outline, the last work that I'm going to present, it's actually a very short teaser presentation, is not even related to imaging. It's almost like my pet project for the last few years. It actually focuses on human speech and understanding how to I'm trying to computationally manipulate human speech. And so if I'm trying to motivate this project from a sort of a brain point of view, here's a very, very abstract view of the brain. So there are sensory input. It goes through some processing of a black box, which is the brain, and then there's some perception. Now, in the projects that I've mentioned, we're able to kind of query the perception with these behavioral assessments, and we're able to query the black box a little bit with some of the neuroimaging modalities. But then the question becomes, what can we do with this sensory input block? And in fact, can we manipulate input-output relationships? So can we change the input to essentially get a different output of perception? And so the project itself, the goal of the project is to automatically amplify emotional cues in speech. And the clinical motivation is from a lot of work that I've done with autism collaborators. So in autism, many of these children have difficulty grasping social and emotional cues, particularly in language, so in speech. But for especially high-functioning individuals, if it's very exaggerated, so think of a cartoon, it is something that they can perceive at the same level as their peers. And so the idea was, well, what if we could design some sort of AI algorithm that automatically could amplify or inject cues in ambient speech to the point where an autistic individual could perceive them. And we can think about using this as a study tool, so designing behavioral paradigms or potentially even kind of an assistive technology. So while this was a wonderful kind of high-level goal, it turned out the problem with it is that no one actually knows how to amplify emotions in speech or even put emotions in speech. So it's an unsolved problem. And so in my perhaps naivety, when I started at Johns Hopkins, I decided, okay, we're going to do this. And so in order to think about designing a model that creates emotional speech or takes speech and makes it sound emotional, well, we need some amount of data to be able to learn this model. So it's a perturbation model. So what we'd really like is kind of many examples of the same person saying the same thing, but in different emotions. And so you can kind of tease apart what is different between the two. It turns out that data did not exist. And so again, in our infinite naivety, we decided to collect it. So we designed a phonetically balanced script, neutral words and phrases, and we actually hired actors in the Baltimore area to come into the lab and read the script in different emotions, well, portraying different emotions. So we just have a few canonical ones here. This entire data set kind of construction and collection was really spearheaded by an undergraduate in my lab, so Jacob, who graduated just recently. And once we have the data, so the speech data, well, beyond knowing what the actor thought they were doing, we wanted to know how people perceived it. And so we took the entire data set and put it on Mechanical Turk. And for every utterance, we kind of crowdsourced it to get different user perception of what the emotion in that utterance was. Okay, so now we have the data to build the model. And the question is, how do we build the model? So if we look at speech, so this is just if you were to plot just a graph of something being spoken over time, this is what speech would look like. I'm not entirely sure what's being said, but it's very similar to this pattern for a wide variety of utterances. So from here, it's not really clear what we would do to manipulate it at all, while making it still sound like speech. But if we squint, the signal looks a lot like random noise with an envelope, right? And in fact, this is how the speech community analyzes speech. So we can take a single segment or an utterance and use a vocoder. So there are a few that tend to be used. So straight is one common one, and then world is an iteration of straight. And what it'll spit out are three components. So the first is a spectral envelope. So you can think of this as a smooth spectrograph. And the second is aperiodicity. So this is kind of the deviation from being a pure sinusoid. And then the third is a pitch contour. So the spectral envelope is very commonly used in voice morphing. So if I want to make speaker A sound like speaker B, because everyone has kind of a unique spectral signature. But for emotion is really characterized by intonation. And so intonation is best described in the pitch contour. So what is the primary pitch over time? So our strategy is to actually just focus on how do we manipulate this pitch contour in a reliable fashion. And then the vocoder allows us to throw those three inputs and it'll reconstruct speech for us. So if we take a look at the pitch contour, so this is the same utterance being spoken in kind of a source emotion and a target emotion. They look very different. So my guess is that the source emotion is probably neutral and the target is probably angry, just based on how things tend to look. The entire manipulation is work done by my graduate student Ravi at Johns Hopkins, and then our collaborator Nicolas Charon, who's in the applied math department at Johns Hopkins. So if we take a look at these two contours, and then we look at it from maybe a computer vision or even sort of a medical imaging perspective, well, the first thought that came to my mind is, okay, why don't we just register them? So essentially, we can treat the problem as a two-dimensional curve registration, where our goal is to kind of find this transformation that'll move the source pitch contour and align it with the target. And in this particular work, we even have one additional simplification, which is we can align the segments over time. So the speech community, there is a dynamic time warping algorithms that allow us to do that. And so essentially this transformation is just vertically, so across frequency. Right? There are many registration algorithms, but we've chosen to use LDDMM for a few reasons. First reason is that these contours are very different. This is a large warping. It's not necessarily a small perturbation. And so LDDMM has the ability to do that. But most importantly, the entire deformation is just specified by an initialization. So the initial momenta vectors, so initial vectors at kind of sort of periodic frames throughout the signal. So it's a very low dimensional representation that specifies the entire transformation uniquely. So our target is to use our training data to learn a function to predict the initial momenta. And then once we have that, we have a standard warping process. So we use a highway neural network to learn the initial momenta. And so the inputs to this neural network are spectral features. So these are features based on the spectrum at a particular kind of time interval t, and the original pitch contour. The network itself is actually fairly simple. These are all ANN layers, but we include some skip connections to have a smooth flow of the gradient. And then used for training, there is actually an optimal control problem given a source and a target contour that we can use to obtain the initial momenta. And once we do this, we can train all of the weights of the network. And then during testing, just as a kind of in an open loop fashion, feed all of the features to predict the momenta and use LDDMM to work. So we have a few preliminary results. So we used the VSIS database. So we had some amount saved for test training, validation, and testing. So these are the results on the testing. We have our method, which is the proposed method, and we compared it to one of the recent iterations in the speech community, which is just end-to-end deep model using LSTMs. And so from a metric standpoint, we're able to do better, probably because we have a very constrained space, whereas the BioLSTM, it was just too rich and it couldn't fit properly on the amount of data that we had. We also ran a crowdsourcing experiment on Mechanical Turk, where again, our model in general is users are better able to perceive the manipulated emotional content from neutral. And then just for fun, I had an example. So there was a little bit of an issue with the projector playing.
Pass.
Yes.
Which floats past.
So unfortunately it cuts off the first part, but this is one actor saying the jellyfish floats past.
A jellyfish floats past.
There we go. And this is our modified.
Past. Floats past.
So there is a perceivable change in intonation. Unfortunately, I can't get
Past.
can't get it to start
Floats past.
at the beginning of the utterance, but sort of, at least to my ear, because we've been listening to this a while, there is a change in intonation, and it does sound qualitatively more emotional. In this case, it was supposed to be angry. What's interesting is that this actor was very expressive. So this is the actor.
Floats past. Floats past.
My students have assured me they didn't mix up the sound files, and it still is the same person, but very different. Anyway, this has just been a fun project that we're working on different iterations. So moving forward, trying to, instead of just time aligning the signals, actually capture changes in durations, because different emotions tend to be faster or slower in terms for certain segments, and also amplifying intensity. So before concluding, I'd like to thank my wonderful lab, collaborators, and funding sources. And so just to summarize, I've presented 3 very different projects that all have this theme of blending old school and new school machine learning techniques or data analysis techniques for neuroscience problems, and also spanning a range of sort of utility and translation from basic science to therapeutic planning to even potentially in the next maybe long term developing new naturalistic therapies. So with that, I'm happy to take questions.